have a saturated Saturday. Yeah, and some of you will be having different timings. So wish you all a happy Saturday. Okay, and then do you remember yesterday's class about the genetics? Yes, I hope you would have understood the concepts very well. Okay, so it's a class 10 test session for you people. Okay, I told you yesterday itself tomorrow will be a review or the test session will be there, so that you will what to say, that you can judge yourself that how far you have understood the concepts, and then how you can recollect that during the examination. Understood? And then how to prepare for the exam also. See, the some of the tips I'm going to give in this video. Okay, and then what do you mean by the term heredity? Any idea? So the heredity means what? It is the information or the genetic information. It can be a physical appearance or it can be a genetical information. It's transmitted from one generation to another generation. Is that clear? And then, who discovered the basic principles of heredity? Who discovered the basic principles of heredity? So in the beginning of the lesson, which I have given as in the video form, that is DNA I have given. In that I have mentioned about the a person or the scientist who have discovered the heredity. So find out the answer and then come back. And next question is 9:3:3:1 ratio is due to, I have given 4 options here. So it's nothing but the segregation, crossing over, independent assortment, and the recessiveness. So this is the ratio. It's a dihybrid cross ratio, I think. Yes or no? Am I right? Mendel did his dihybrid cross ratio by crossing 2 or more triads. Yeah, yellow with the round and the wrinkled seeds with the green. Yeah, so the 2 or more characteristics have been selected by Mendel during his pea plant experiment, and he derived the ratio 9:3:1 ratio. Yeah, and then this answer, what could be the answer for this ratio? You can comment me if you know the answer, so that you can test yourself how far you have learned the genetic concepts. Yeah, then next question is, I will discuss the answers, don't worry. I'm just first giving the questions to you so that you will think and you can come back with your answers. And the next question is the units, the dash units which form the backbone of the DNA. Here again 4 options have been given, which is the backbone of the DNA, whether it is a 5-carbon sugar or it is a phosphate sugar or it is the nitrogenous base or is a sugar and the phosphate together. Yeah, please find out the answer for this third question also. And then the fourth question is, what is the number of chromosomes? What is the number of chromosomes found in human beings? They are, here again 4 options have been given. So what are they? They are the 22 pairs of autosomes plus 1 pair of allosomes. I told you that autosomes are nothing but the growth chromosomes and allosomes are nothing but the gender or the sex chromosomes. Is that clear? So now the next option for this question is, I have given, please wait. The next option for this question is, the second option is 22 autosomes plus 1 allosomes, and then 146 autosomes, and the last one is 46 autosomes and 1 pair of allosomes. So from these 4 options, which is the right answer? The next is Fifth question is Okazaki fragments are joined by dash. How are they are joined? They are nothing but the segments. The fragments are nothing but the segments. They are joined by the which enzyme out of this? The helicase enzyme or the DNA polymerase enzyme or the DNA primer enzyme or the DNA ligase. So which is the right option for this question? Yeah. Shall I repeat the questions again? First question is who discovered the basic principles of heredity, and then 9 is to 3 is to 1 ratio, 3 is to 1 ratio is due to which option, whether it is segregation, crossing over, independent assortment or recessiveness. And the next is the units from which form the backbone of the DNA. They are either the 5 carbon sugar or the phosphate or the nitrogenous base or the sugar plus phosphate. And the number of chromosomes found in human beings are dash 22 pairs of chromosomes. They are autosomes, which are nothing but the growth chromosomes, plus 1 pair of allosomes. Allosomes are the gender or the sex chromosomes. Then next is second option for this question is 22 autosomes and 1 allosomes and 46 autosomes, and then 46 pair of autosomes and then 1 pair allosomes. Which is the right answer? You can please comment me if you know the answer. The answers will be posted in the community. So the Okazaki fragments are joined together by which enzyme? The one is the helicase, the another one is the DNA like polymerase, and the RNA primer and the RNA ligase. And we have discussed about what is the term Okazaki. The Okazaki means it was the name which was named after the scientists Reiji and Okazaki, who found these fragments during their study in the E. coli DNA fragments. Yeah, so that fragments were named after their name. And here which is the answer for this question? Please think, please come out with your right answer. So not only this multiple choice, I am going to share with you one higher order thinking question also. Please make a note of it so that the next session you can come out with your answers, and then I will also post the answers in the community. Okay, so look at this question. This question is so second question I am going to concentrate here. Here the pure breed tall pea plants are first crossed with self-pollinated, with a pure breed dwarf pea plants. Okay, I may repeat. Pure breed tall pea plants are first crossed with a pure breed dwarf pea plants. What do you mean by dwarf? They are the short. So you have to recollect the Mendel's experiment. What he did, he crossed the tall plants with the dwarf plants. Am I right? Yeah. And then the pea plants which are obtained in the F1 generation are then self-pollinated to produce a F2 generation of pea plants. Okay, so just recollect the Mendel's experiment. So in the first generation, Mendel crossed the tall with the short, and then he got some plants in the F1 generation, and then again he self-pollinated plants which are produced in the F1 generation. So and then he produced the F2 pea plants, F2 generation. Understood? So now what is the answer for this first question? What do plants of F1 generation look like? In the F1 generation, he got some plants, no? They showed the similar triads, similar characteristics. So what is the answer for the first question? You can please comment if you know the answer. And what is the ratio of tall plants to dwarf plants in the F2 generation? And then which type of plants were missing in F2 generation but reappeared in the F2 generation? In the F1 generation, it is reappeared in the F2 generation. Clear? So I repeat the question again. Pure breed tall pea plants are first crossed with the pure breed dwarf pea plants. The pea plants obtained in the F1 generation are then selfed to produce the F2 generation of pea plants. What do the plants of F1 generation look like? And what is the ratio of tall plants to dwarf plants in the F2 generation? And which type of plants were missing in the F1 generation but reappeared in the F2 generation? This is the higher order thinking question which I have given you. So please think, and it's the answers are simple. I hope so. You can come out with your answers. Okay, so the answers will be posted. Yes, and then now we will discuss again the recap of the genetics. Okay, yeah. So I will show you the concept map so that it will be useful for you to recollect the genetics again. So next, so look at this concept map. The concept map will show you what is the chromosomes we studied in this lesson, and what is a DNA structure, and what is the mutation we studied, and finally Mendel's law of heredity also we studied in this lesson. Yeah, so first and foremost is Mendel's law of heredity. So Mendel conducted experiments in the pea plants, no? So what are the experiments he did? He did the monohybrid cross, selecting the tall and short plants, and he also did the dihybrid cross, selecting the two or more characters together, that is yellow and the round plants with the green and wrinkled plants he selected. And then he came to a conclusion that there are certain things which what to say help his experiments, and he found the results with the discovery of laws, so that they are the law of dominance, we can say. So the law of dominance and the law of segregation and the law of independent assortment. So for all this I have given a video stating that segregate, assert, like that, dominate. So that video topic will be like this, stating that segregate, dominate, and assort, independent assort. So you can go to that video. I'll share the link with this. Okay, so that it will be very useful for you to understand. Okay, now next we studied about the DNA structure, isn't it? What is the DNA structure? DNA is made up of, what are the basic things present in the DNA? Basic things present in the DNA are, that is, DNA is nothing but a polynucleotide, yeah, which is many in number in the living organisms. And what are the parts of the DNA? DNA has a pentose sugar, DNA has a nitrogenous base, and DNA has a phosphate sugar. So a pentose sugar is a ribose. Okay, what is a pentose? It is a ribose, and nitrogenous base are purines and pyrimidines. And then the phosphate group is the phosphoric acid group, and it is the phosphoric group is giving the acid character, acidity to DNA. Hence the DNA is known as the deoxyribonucleic acid. Am I right? That's why the nucleic acid, we say it is containing an acid, is mainly because of the phosphate group, which will provide the phosphate. Phosphoric acid will provide the phosphate group. So this will act as the donor of proton H plus. That's the structure of the nucleotide, and then that is DNA nucleotide. And the next we studied about the replication. Okay, so the replication, first step is origin of replication and unwinding of DNA, and then the formation of DNA primer and synthesis of new complementary DNA, and the termination. So in the origin of replication we studied, what did we studied in the origin of replication? We studied about the site, which is a specific site the DNA will select in its structure, and then it will open, that is the unwinding of DNA, by forming a replication fork with the help of the enzymes helicase and topoisomerase. Do you remember? Yeah. And then there's a primer, RNA primer, which is also formed by the DNA by itself, and this primer only will going to initiate the replication process. From there only the DNA will start producing the new strand to the template strand. The template strand is called the parent strand. Okay, so the next is synthesis of new complementary strand to the parent strand. And a particular site, when the DNA think whether that is the replication is enough, it will stop its replication process. That is the termination process. Clear? And the next is we studied about the chromosomes. So the chromosomes are nothing but the group of genes. What are the genes? Genes are nothing but the group of DNA, I can say. Yes. So next is we studied about the different structures of chromosome. So for all these structures, types, and the kinds of chromosomes, I have posted the video. I'll share those link with along with this live session. Okay. So it will be very useful for you to recollect the concepts again. So which I have explained in Tamil also for those who are studying in Tamil medium. Okay. The next is so what is the structure of DNA? DNA consists of primary construction and then secondary construction. There will be a centromere, telomere, and the satellite. Yeah. So what is the primary construction? Primary construction is this. This is the primary construction. If there is a link between this one arm of the chromosome and another arm of the chromosome, this is the primary construction. And there is a secondary construction here. This will link the arm of the chromosome with a bulb known as the satellite. Okay, this is the secondary construction. And the next is here also we can see in the another arm of the chromosome we have a secondary construction that will connect with the telomere. This is another part of the chromosome. Apart from that, your chromosome will also have pellicle, matrix, and then all other parts, spindle fibers, the chromonemata, everything will be there, and this is the structure of the chromosome. And then we have studied about different types of chromosomes also. So what are they? It is the telocentric chromosomes, and then acrocentric chromosome, and then the submetacentric, and finally metacentric. So for all this I have posted the video explanation, so please go and click on the links and then you can learn better. Okay, and the next we are going to concentrate in the next concept is it is the that is kinds of chromosomes. I already told you there are autosomes and then allosomes. Autosomes are nothing but growth chromosomes. Okay, growth chromosomes, and then allosomes are nothing but the gender or the sex determining chromosome. So how many pairs of chromosomes we have in humans? We have 46 pair. In this 44 pair is the growth, both male and female, and one pair alone will be the gender determining or the sex determining chromosome. In male it will be XY and in female it will be XX. Yeah, we have studied about the sex determination also, no? During the zygote formation, how the DNA will be splitting and how the number of chromosomes split from each other. So this 44 and 44 will split into 22 plus 22. From the male you get 22 plus 1 chromosome, sex chromosome, and the female also you will get from 22 plus 1 sex chromosome, and they again rejoin and you will get a male or a female. It's a probability, we cannot predict whether it is only exactly male or exactly female, we cannot predict that. Yeah, so for sex determination, gender determination also I have posted a video, a link also. So everything I will share with this, so you go through all the videos and you will understand better, and then you can answer the questions. So the answers will be posted in the community. Okay, so thank you so much for coming and attending this live review session, revisit session for the genetic topics. Yeah, so we will meet in the next live class. Okay, and then answers will be posted in the community. You can please check and you can also come out with your own answers by your comments. And I want everybody to give your positive as well as the negative comments. If there, please let me improve myself. Okay, and we will meet tomorrow in the next session, or if the live session is not there, I will post a video also. Okay, you can check my videos. Yeah, thank you so much for your cooperation. Wish you all a very saturated Saturday. Thank you. See you. Bye.